work with you and also your interest levels on these things. I do want to get through the Archie, and I don't know where the semester goes. Usually I have, it's like we're 1 or 2 weeks short. I usually cover a bunch of other, smaller topics at the end, and we just don't have it this term. But I'm happy to have gotten through all the diversity. That is important to me, and I think as a biology major, not a microbiology major, it's good to study diversity. So we're going to do that. If we have time, somebody said, we're going to do a review tomorrow, aren't we? Okay, so can you do the Jeopardy thing? All right. So I got before and I put something together on that. I think it's going to be kind of like rapid Jeopardy if we do it, and we'll see how much time we have. And are you interested in that? Do you want to do that again, or is that like, I've done that?
Definitely.
Huh? Definitely? Okay. So, and I'll make it available. Even if we don't get to it, you can read all the stuff. There are questions right off the test, so I think it's a nice way of getting a flavor for that. And then the last thing, or maybe the first thing we should do, is the class evaluation. I think you guys kind of know the score on that. Evaluate me as an instructor in the course and the way it's laid out and what's covered, and your satisfaction or dissatisfaction with that. I know I got behind on correcting things. A lot of professors, when they get to be full professor, they kind of go to pasture, they burn out. I try to stay involved with service, such as giving a show in St. Louis, and research, but it does, it's hard to juggle it. But you can also say, from your perspective, I'm paying good money, you should, I should be first. So I get it, but so it's certainly hours in a day. So hopefully it wasn't an egregious error, but perhaps, no, just let me know, just write it down. So that's it. Oh, the test, 8 o'clock on Tuesday, so come early, just because some years, because I had tell people, there's no time when it's over. People trip in like at 10 and start the test. I hope we can all start at 8. I mean, don't take advantage of my good-naturedness. I want you to have time, but don't trip in at 10 and expect me to stay to 4. Start at 8, you want to stay to 4, okay, I'll sit with you, that's cool. But, meet me, at least at the starting gate, okay, on that one. Assignments. You like it? Okay. So, I would try, today I'm going to go and set up, try to stay and correct things tonight. I will post as things get corrected. The big thing is, if you're really on the line in this class, you can write to me and say, can you correct my stuff first? I'm really worried, and I want to proportion my time, and I'll try to meet you on that, okay? Some of you, really, one end or the other, who knows? Probably not a big thing, but if you're really like, well, if I don't have to take that comprehensive, I can spend a lot more time on that other part of the test. Okay, fair enough. I get it. So let me know on that. I will go through and correct things in batches with the reports coming first and presentations. I'm really looking forward to a lot of those. And keep you posted. If you're sending stuff to me, you miss deadlines. I don't know. There's all these things people do. I know that'll probably come second. But if you don't see your grade there, say, hey, I sent that to you. That's fine. And even when we're done with the final, I hope that everything wrapped up by Wednesday or Thursday. So if you don't see stuff, just say, hey, you missed that grade. Okay, if it makes it into Blackboard, I find it. If it gets sent or shoved on my door or something, you've seen my office. I do my best, but it's definitely a detritus pile. So, okay, questions on that stuff? I think we kind of covered most of it. All right. You want to do the course evaluation now or at the end? Now. Okay. Can I have a volunteer that will take this to the proper receptor domain that you may have learned about a few years ago or in other textbooks as the archaebacteria? But people say, well, no, there really aren't bacteria. Let's not confuse these. Okay. You may have also learned about them as this idea of extremophiles. I got some extreme things going on here, but and to some extent that's true. Extreme salt, extreme anaerobia, in your intestines. Extreme hot is a good way of saying that's where people started first noticing. Okay, and like I think I've told you, Sarah Paver and a few other, some of the CAP scholars got real interested in shadowing us in our work on Illinois River, and they started saying, well, go see what the archaea are out there, and there's a lot of them. And it's not unusual. Now we see that they are major players in the nutrient cycles. They're not just these obscure things, living out, in the geyser pools and stuff. So our picture is changing. I also told you about the deep sea explorations about 2 years ago, where they may have found whole other groups that are really like the archaea. So the story is still open. Okay, it's a fascinating, weird world out there. So we're in this part of the tree. Here is Luca, whatever that might be. In some ways, and we've gone through the cell biology. I'll touch on it lightly, but it doesn't hurt to maybe look at some of that stuff early in the class. We laid down that foundation for a reason. If you look at things in some ways, we are more closely related to them than we are the bacteria in some of the gene overlap and some of the biochemistry. But as we also see, well, it's a pretty far rock to throw on that, so that's fine. Okay, so we're in this domain. The diversity of the archaea, as I mentioned, are changing. Probably when we started out talking about these, and we'll emphasize these today, we're talking about the Euarchaeota, okay, the true archaea, okay, the Crinarchaeota. Don't really know what that means actually. I've never looked that up. The Crinarchaeota. And to some extent, the Chordaarchaeota were in the mix a few years ago. The microbes that we think came from Mars kind of fell in there. Like, oh, they came on asteroids, stuff like that. A little bit, this has been defined a little bit more. This one is kind of out there. The Nanoarchaeota is good. We're just going to touch on. They're starting to find things that just didn't quite fit in these other ones for other reasons. We're not archaeota scientists. We won't get into the subtleties. We're going to be doing this kind of tour on our way down Route 66 of the bacteria. Okay. It's an interesting domain, okay. We see more in here because we know more of these, and they're probably in areas that we're exploring more. We find we initially knew more about microbes in us than we did in, on plant roots and things. Okay, the Crinarchaeota about the same way. You'll see some things like, wait a minute, you told me that nitroso prefix really went with the nitrifiers. What were you saying? With the bacteria, that's true. We start to see it now. I don't name these things. We start to see it coming up again in the Thermoarchaeota because people are finding that the archaea do some of the functions that the bacteria do. What I told you is not a lie with the bacteria, but I tell you that this now so you would not get confused looking at it. You would have to look at it again. In our class, we're not going to emphasize that too much. So I kind of talked about that. When we get into the Crinarchaeota, we're going to see thermo and sulfur a lot. In the Euarchaeota, we're going to see methano. That's a big deal. We talked about the methanotrophs, okay, those that eat methane, okay, trophic. Now we're going to look at methanogens, genesis, the creation of methane. Okay, so we see those guys in there, and we see some halos, some salt ones, and thermos always with the archaea. They just seem to have been here since the beginning of life. This is just another version of that emphasizing the Euarchaeota and Crinarchaeota. I like things like this. I'm a pictorial learner, so for me, seeing things mapped out, and if I just sit there and kind of look at it for a while, it tends to make more sense to me. So it's a an aid for you in that. I'm not going to dwell on it. We've talked about different parts of the cell biology. Like I said, I'm not going to emphasize that too much because we've studied it, you've written about it, you get it, but I want to remind us a little bit going into it. That's when the archaea first started to be discovered, besides being extremophiles, they said, well, they're prokaryotes, so therefore they're bacteria. And there's a lot of controversy about calling anything a prokaryote now in some of the circles, but let's not go there today. But so people say, well, you got the eubacteria, the true bacteria, and you still see that. And then you have these archaebacteria, the archaean, the older remnants, ancestral bacteria. And now with these types of things, people say, we just got to call them something else. And then they made the names of the domains as you know them. Where did they first discovered? Yeah, we talked about Thermus aquaticus, a bacterium that was found in these obsidian pools in Yellowstone, or octopus pool. I have an obsidian pool, that was a very insightful thing. Well, let's start dipping in this hot stuff and finding it. Now, I want to just tell you, as an environment, as an ecologist, it might be, well, it's going to be over 100 degrees. These are mineral springs, right? You pour salt into water, it boils at a higher temperature. But there's also gradients that go on in here. So, they had to go back and kind of fine-tune some of the conditions. You just stick a thermometer in the middle of a pool. It's real different when you get down to the level of, where a bacterium, or an archaean can live. So, I just realized that the diversity of habitats open to something that's a few microns is real different than if, you and I decided to jump into an obsidian pool. So, okay, we're going to talk about in the Euarchaeota, the true archaea. They're no truer than anything else. It's just, I think they're the first group that they named, so it's not like the rest of them are false archaeota. We're going to talk about halophiles, methanes, and talk a little bit about some of these type of thermophiles. We've crossed that bridge before, but now we're going to deal with this whole different group. Some people in the lab looked at halophiles. They really are cool. We see these orangish salts that come from the Himalayas, and we thought, oh, maybe we can get archaea off there. We weren't that lucky, but where did we get that off of? Sea salt, right? Weren't you doing the salt thing? No, who's doing the salt? You guys got them off of sea salt, right? So that came, it doesn't mean they're not there, just that we couldn't culture it in 2 weeks. Well, we were asking a lot of that, but this isn't just a salt crystal. Some of them will actually have a square shape, something we don't talk about too much. Oh, those are the square ones. Yeah, and then we'll see some of the other, mainly rods and so forth, but that's kind of cool. We're talking about things that require 1.5 to 4 molar. I think our group decided to go test the upper limit of that, and that's really asking a lot to grow something above 4 molar of anything. Okay, it's a real test of the osmotic condition. Here, some of you have a lab book that shows some of these salt flats where people will dry saline water and get crystals and gather salt and sell it or use it in their towns or whatever. And it'll start out getting real salty, and as the bacteria grow, they start to develop, it looks like they're turning red. And at first you go like, well, what's going on there? Is that another mineral? It turns out that halobacterium, kind of our poster child for the salt, the halophiles, have bacteria rhodopsin. Okay, rhodopsin is a light pigment. So as they start to be able to lose or use up their organic substrates, they start to shift over to not a really well-developed photosynthetic pathway to gain energy. Okay, you have rhodopsin, right, in your eye. Do you know that rhodopsin? Do you know the sneeze trick with rhodopsin?
You look at light.
Yeah, you look at light. Yeah, so you ever get that? You can't sneeze. don't look at the sun, don't do that, I'm not saying, but if you can look at some, bright light, rhodopsin, and I don't understand it, maybe the biochemists do, but the rhodopsin gives, it tips over the sneeze response so you can balance your CO2 and all that kind of stuff, but that's rhodopsin. That story you might know about yourself, and it's a fun trick if you have to sneeze a lot. So we do, we're not going to get into this today, but we do have these pigments that are in the membrane that are able to acquire energy, make ATP from light is the main lesson on this today. So, where can you find it? Great Salt Lake. I was out there. We went and got some salt water and looked for some halophiles. You don't have to go to the Great Salt Lakes. You can find halophilic bacteria in salt mines. In Lake Onondaga in New York, they found halophiles, and it turns out that it came from, there's no, Lake Onondaga is a very polluted lake in New York, highly studied. It turns out that the halophilic bacteria actually came from Utah. People were importing salts and salted meats and dumped the salt, and around Lake Onondaga they started picking up halophiles, which is kind of interesting. Okay, the big thing on this one is how do you maintain life when you basically have all the water sucked out of you? Okay, 4 molar salt is high. Do the math. You guys can do it. 58.4 grams per liter. That's a lot of grams, like 200 something. Basically they have a proton pump, so they pump large amounts of potassium in, okay, and they get the sodium pumped out, okay. So that in itself changes a little bit of the osmotic stress, but also balances the osmotic stress in the organism. And so they make a greater amount, they're able to not have such a large chasm between the outside osmolarity and the inside osmolarity. It's still a lot of salt. It's still a lot of stuff going on, but they cope with it by balancing protons. Talk about that. Okay. We're going to move on. As I said, we're kind of doing a skip, hop, and a jump through the whole domain, but you'll get the idea. I love talking about the methanogens. My first graduate student just wanted to study the archaea. We did all this, PCB work and so forth. I get a kick out of methanogens. So our thing here is looking for the prefix methano. Not hard. Okay, get it. Methano. And then look for methanococcus, methanosarcina, methano, whatever the rest of the word might be. Now, here's Volta. Okay, this is a smart guy, he figures out volts. He's a physicist, kind of a Renaissance type guy. In the 1776, we're doing other things like trying to get our independence in this country, and he starts to think about this kind of thing. Here, these people are in a swamp. This could be in Illinois, it doesn't matter. If you've ever been in a swamp or you had to go in one, maybe you had to do some wading, you smell this sulfur thing. You think, oh, is the sulfur from the methanogens? No, you can't smell methane. But what you can do, these guys went through, and as if you were to walk, I used to do this with students, that if you walk through a wetland or a swamp or something, you'll see bubbles coming up, and it won't smell so good. I usually just get a big garbage bag, and I collect the bubbles. Okay, these guys used a big Erlenmeyer flask. You can collect all these bubbles, and kind of fill up, at least partially, the garbage bag. And then what we do is usually gather the end, make a little orifice, and then someone gets a lighter, someone else pushes the gas out, and you get this. Okay, you get methane. Like, wow, where'd that come from? Well, it came from the archaea. Okay, some of us produce methane. This is it in a nutshell. The big deal about the methanogens is that you have to work under highly reductive habitat. Okay, so those of you who are soil scientists, you're working at minus 400 millivolt reduction potential. The rest of us that were sending like nothing to, we're talking about no air, zero air, not even a hint of air. Okay, because you're taking this wonderfully balanced C double bond O kind of thing, and you're going to reduce it down to its most reduced form. This is tough. Okay, I mean molecularly tough. Oh, that's not. Now, this is the basic formula. Just like we have the basic formula for photosynthesis. There's variants on this theme, but I wanted to blow this one into your brain to kind of burn that in. That's a big deal to oxidize that. It turns out that almost any other electron acceptor in the soil will be used first before those that can do this will do it. Nitrate, iron, manganese, sulfate, all those by the other consortia will be used first before you get to making methane. Okay, they don't look like a lot. No really cool internal membranes and that kind of stuff. So they're obligate anaerobes. Talk about where they're found at. Okay. Yes, CO2 is fine. These guys, just like the methanotropes, are working within the cycling of what we call the C1 metabolic pathway in the carbon cycle. Not hard to figure out because methane, one carbon. Okay, other things can work out. Now, this one, formate, not going to get as much, of an exchange. Anything that has a methyl group, methanol works well, and even acetate with 2 carbons will work. One of the things we used to do in advanced micro is we'd set up methane digesters. We'd get mud. The best mud comes out of Kilbourne, Illinois, as far as I can figure out. I don't know why. But how I found that, I don't know. But I found that's better than anything we found. And we set up these digesters, and we figure out the molecular equivalents of these, and we set up different digesters with the carbon. And I know you can, the chemist can sit down and figure out the stoichiometry right away, but generally, most people don't. I don't tell the students they can do that. And we take bets. This is like we bet a lot in advanced micro, the $50 on the Winograd-Ski, $20 on this. Which one do you think is going to produce the most methane? And we try to we figure that out, just to get the fact that you can have different methane producers from different substrates. In here, coenzyme F420, really, you can take mud and put it underneath UV light, and you'll see they'll glow. And it's because we're sitting with a UV wavelength off this enzyme. You really have to memorize that. I guess just kind of know that one of the main enzymes is fluorochromic, but other than that, probably a lot for the end. Oh yes, this person's very happy to be working with acetate. I just had to put that in to share our day out. Actually thought I took this slide out. When we have, why is glucose important? It's not that they're going to metabolize glucose, but when these leaves, when trees, when things are decomposing, in aerobic soil, in aerobic water, they break down fairly fast by a different pathway. Anaerobic breakdown, like a lot of you did with Winograd-Ski column, is starting with Clostridium, starting with some of these other anaerobes, and you're fermenting. So you're not only putting out ethanol, but you're putting on other small fatty acids and things that are slightly reduced, and you start to get a pathway that feeds into it. Not as great as with air, fair enough, but it's just not like, well, they can only use CO2, and it's only going to happen in mud and stuff like that. How long was this class last? Oh, wow, okay. I'll talk slow. No. Here, the red, what is it, a rectangle, is the methano thing. Okay, there you go. Lots of different names at the back of them. cool. It's fun to do the methanogen. When Kurt was doing his doctorate, he went to a special workshop to learn how to do it, and we had to pump in. So when you do it, if you don't have an anaerobic chamber, you have to pump in gases. So we had methane being pumped in one, and nitrogen in the other. We have these big cannulas that are in the tubes and so forth. And all the faculty at Pace, now I didn't get kicked out of Pace because of this, but all the faculty at Pace thought we were going to blow up the whole place. We never did, okay? But you can grow these things. You can do these things even if you aren't at a multi-million-dollar institute. Okay. In this table, the other things, okay, do we have different shapes? Sure, great. My favorite Methanosarcina down here, I just love the way that one looks, but we've talked about that. This is just to show you some of the diversity of these substrates. Here's formate, here's CO2. Nothing that we haven't really talked about before on that, but just to realize that you can have a lot of different products that these guys take a hold of, okay? And not always at the spectrum of going down to CO2. And we saw it in the last one, so we can have not just, we can have other derivatives of chemicals. That's the C1, maybe a C2 are the big deal with these guys. Demonstrates the diversity of cell wall chemistries. This is an FYI. It's hard for me to be general about the archaea. You say, well, some have a protein S layer, some have this type of cell wall, that type of cell wall. Here, even within groups, they seem to have different types of constructions. So it's not like that kind of thing, like, oh, it's an ester linkage, you know, and an ether linkage is good for all the archaea, but when you get past that, it's kind of like when we were talking about fatty acid composition in the bacteria. You started to see, we talked about in general, when we talked about FAME, oh yeah, we got the basic fatty acid moiety, but there's a lot of diversity within it. That's how I think you need to think about within the groups here. Not that one size fits all, that they do have some of these different characteristics for their cell wall, is my point on this slide. Cool looking. FYI, I will go quickly over. I'm just talking about Kurt Jerke, great guy in the armed forces, but in between Afghanistan and all those things, did a really nice PhD. Our idea was, I told you the story about the cattail. So here's my cattail root. And I said, well, around the cattail, there's oxygen. So we have this oxic zone. So my idea was that between the sulfate reducers, which we've talked about, which will take off certain chlorines, and the methanogens, which can take off other chlorines through reductive chlorination, we could reduce and get rid of PCBs, which we were working in New York, the Hudson River, full of PCBs. We did. We went to Fordham University at the Caldwell Center. We filled up their greenhouse with PCBs. We kind of got kicked out of there. But we did it. It was able, we were able to dechlorinate using this kind of model. So this is an FYI. You can think about the microbes in a different way. Okay, Julie Mavis took our course, looked at microbes, and understood bioremediation, started her own company in Illinois, and used some of these tricks that we're talking about. So that biochemistry you're learning, along with physiology and microbes, can really address some really important questions in our world. One place where we bring together several different ideas is syntropy. We talked about that before. Basically, you need two different organisms to attack and bring something to a different metabolic end. Okay, nutritional that's in. In cellulose, we have that kind of thing going on in the gut of termites and things like that. So we have cellulose being fermented into sugars CO2 and hydrogen. The problem with hydrogen is it tends to be a competitive inhibitor for the next stage. So what you want to do is get a centrifugate and get rid of the hydrogen so the methanogens can come in and push the fermentation further is the deal on that. I know I'm going through that a little bit fast, but the big idea is that you got to have two different groups working simultaneously in order to push the reaction forward. So yeah, centropenols not grow in the presence of hydrogen, so it stops it. Okay, we say, well, just get rid of it. Well, if you're in the soil, it's a little different. It's funny, because hydrogen is kind of limiting. But the methanogen says, yeah, give me some of that hydrogen. That's cool. I'll take it. And it opens up these guys to continue doing their work. This kind of looks like a funny little Wookiee or something. It's like Chewbacca the microbe. What we have here is a ciliate, and this is actually the archaea on that. They're getting acetate off of the ciliate and being able to metabolize it. So in advance, we talk more about all of that. It's pretty cool. Okay, in the Yoda, we do have some things that like high temperature and extreme pH. So I throw some of that in there. This is the pH award, low pH, pH 2. Imagine these things would be just fine growing in your stomach. They don't worry about that. But that kind of pH situation, they don't look like a lot. You wonder how, what's the significance? I think it was just life trying to be life. I don't know that they were trying to get the award for growing the most and the biggest, okay? Here, Picrophyllus. People always say, well, how low can they go? 0.7 pH? Whew. Okay, imagine picking up your hydrochloric acid bottle and finding bacteria growing in the bottom, like, huh, where did they come from? It'd be that kind of thing. What do we get from this? The extremophiles are so interesting because it takes us past thinking about the physiology of our bodies, of our cells, and we look at Deinococcus. How do they fix their DNA? We look at some of these other adaptations, Oh, they have kind of an antifreeze like the fish do in Antarctica. They're able to use glycol to stay in cold water. We get ideas from these kinds of things. The enzyme in your Tide detergent is a cellulase. Okay, the way that you can get whiter than white, you can't. I mean, white is white. That's advertising. But what they're saying, I think, is that if you get a white shirt, or a t-shirt, or whatever, but as it wears, it gets kind of frizzy and so forth, and so that frizz doesn't make it look nice and really crisp and clean. You put a cellulase in there, you can dissolve that stuff really fast. Where do they come from? Extremophiles. It's a different world. We get ideas from these things. I don't have a lot to say about thermoplasma that I haven't said ether linkage. For your information, I think we can move on to, they grow in hot places. Okay. Hypothermophiles, Pyrococcus is one talked about actually in the first lecture, so we come back kind of full circle. Pyrococcus for a long time kind of got the award for living at the highest temperature, 106. So go home, boil some water, make some tea, think about jumping in there and living. Not going to work for you, okay. These guys, yeah, it's okay, that's good. How did they do that? I think in the end, I rearranged some of my slides, but we'll come back to some of the adaptations for that. How did you do it? I mean, you were saying, wait a minute, I did PCR with Dr. Bonnicum, or maybe I will, or maybe I did in general biology. We melted or pulled apart DNA at 94 degrees. What's going on here, Lemke? How can they do this? And it's a really, really good question that we'll try to address to some extent. So. Hot vents, these guys are sulfur, kind of like Thermatoga that we were talking about before, and they like to go at high temperatures. Archaeolobus, I just throw in there, is in the same group. Isn't really a lot to look at. The point is that a lot of things can grow in these hot vents, and you think, why? Well, I mean, it could be that these hot vents were very similar to early Earth. These guys that was their refugia. You know, it could be they co-evolved. That's why I asked you on the test, how do you know? You say, well, some of these things just lived here since early Earth. Well, how would you know? How would you know? If they just evolved since, or they're there since the beginning? What would be your idea on that? You can answer this. What I would do is I would grab the 16S and I would see where it comes off in the tree of life, because the tree of life gives us an idea of evolution and continuity, you know, that might help answer that. That would be my first approach in sequencing, you know. In a few years, you'll be able to get your sequence kit in your Morning Post toasties. I mean, it's not that hard to do that. Okay, I'm going through Nigrospira pretty much. This is the Archaea file that I said we were going to just kind of brush over. I do want to spend a minute on the Nanny Archaeota. I like them, they interest me, so of course I'm going to impose my interest on you. But let me tell you why. They're very small. Okay, so you know, we just talked about mycoplasma taking the award for the smallest one. Well, the Archaea, 0.4 versus 0.2, we're not really talking about a big difference there. So I like them about that. And this, to me, is kind of that gateway to that deep sea exploration. Most of those new alleged domains are really small organisms. Well, they're small, who cares? There might be some secrets. How do you live there? What are you doing? Where did you come from? We don't know how this stuff all fits together yet. We learn a lot from it. Even though small genome, it is able to metabolize, but it's host dependent. So we miss things when they're mixed with other things. So Nanny Archaeum equitans, one of the smallest cellular organisms, was thought to be an obligate symbiote to the Crinarchaeota ignococcus. Okay, so here we see the fluorescent Nanoarchaeota around ignococcus. I don't have a big, deep-seated thing to tell you about this, except that this is a world that's opening up to us that we don't know anything about. Now, should you drop everything and go study that? I don't know. You may become broke. On the other hand, if you're an explorer, you know, these are the areas to explore. The exploration exists around us. We don't have to fly to Jupiter. You know, I mean, if you can't, go for it. Buy the ticket. I'll come and visit. But you know, we can still be an explorer. You know, like Magellan or Amelia Earhart or something. You know, we can do that kind of stuff in this day. And I think this is kind of the gateway to those things. So I'm off my little soapbox, but I do think it's pretty good. Chorioarchaeota, root of life. People start, you know, fossils on these meteorites, things coming from Mars. Some of you guys talked about pamasperma, you know, how we're inoculated. Nice stuff. I mean, you did a good job. I don't know. You know, a microbial fossil is hard for me to understand because it kind of looks like all the other little divots in the rock. So I just got to say, this one's kind of that catch-all. It's out there. We do have some organisms that are being put into this phylum of the domain. I would just say keep your eye on it if it is an area that you find interesting. So they're cool looking little things. We're going to kind of move on to our other phylum for today, the Crinarchaeota. Again, I wish I could say, well, these are all the hypothermophiles. These are all the sulfur guys. Probably more so, but we just saw hot guys in the last one, Pyrococcus and so forth. So, you know, you say, well, why? it is, you know, we have one ruler. We're using the 16S ruler to line things up. You know, we take the physiology ruler and go like, well, it kind of lines up differently. Again, my hypothesis is that if we classify things through thermodynamics, we can resolve this. I don't know if I'll be able to really make a big headway into it, but either way, that's another unknown pathway. So what do we have here? We have some extreme hot guys, some hypothermophiles. We've seen that again. We'll talk a little bit about some of the adaptations. We also see extreme cold ones, psychrophiles, things that can live in Antarctica, okay, -2 to 4 degrees. I mean, imagine instead of taking your Petri dish and putting it in the 25-degree incubator or the 35 incubator, your incubator is the freezer. And you put it in there, and most people go, well, everything's going to die in the freezer. That's where I put my spices and my meat and tomatoes for late winter. Okay, yeah. You know, that's what we do, because mesophiles go like, well, that working for me. Okay. These guys, you know, imagine putting your Petri dish in there, you know, you bring it out, you got colonies growing, coming out your, that's the kind of deal these guys can do. I'm not saying that they're going to grow anywhere as fast as a mesophilic bacterium, but they will grow. They're able to do that. Isn't that interesting? So you have to think, what's the barrier to that? One of the things is why I said that cell wall, that cell membrane is an amazing component. If you make the cell membrane with fatty acids that pack together really, really well, if they're saturated fatty acids, it doesn't work so good in cold. So you want to get ones that are more oily. On the other hand, if you're in a hot spring, you might want to have cell membranes at room temperature that are kind of stiff and more like grease and lard, you know, to just make an analogy, that so when you put it in the hot, you can get things in and out of it. It's more mobile that way. Same thing with your proteins. Well, these guys, we have chemolithotrophic bacteria in the Crenarchaeota. Some are chemoautotrophic. We don't see too many phototrophs here in these guys. Here are the two main groups. I want you just to have some idea about, they have sulfur in their name, so it's not like really hard, except for the sulfa vibrio, which is a bacterium. We haven't really jumped up and down on that. Sulfolobus is one. This is one that they showed that picture in that video really briefly. They showed that animation of these kind of strings floating around in these hot pools, hot springs. They do this. They grow within, they put out a lot of extracellular material, and this isn't dehydrated like from TEM. That kind of makes these kind of webs in there. Now they're not thick and heavy and stuff like biofilms in fresh water, but they still do that kind of thing to suspend themselves in an appropriate place. The Sulfococcus. Here we're getting really close to the organism that is at the top of what things can grow at 113 degrees. I just love, the kids have these little fire bacteria, you know, and here are the mussels, right? These are the mussels on the stalks growing at the bottom. Here's these very good. I thought that was pretty cool. So these guys are getting close to the top of the list of what things can grow at, though, that we have found. So, this is a summary table where they're found. Some things there, they don't look all that interesting, and I want to get to this. So, I want to talk about where things live as we think about diversity of life. You know, in ecology, we say that there's a that diversity is indirectly proportional to latitude. So as you get closer to the equator, there's more species of things. Okay. We see other kind of things. You know, we've talked about, or in ecology, you talk about Bergmann's law, Allen's rule, that kind of thing. But you know, the species thing, and people have asked about, well, do bacteria show the same thing? Yes and no. Where do you think the highest diversity of bacteria is per whatever square mile on Earth? And it's not you. You have a lot, but you're not it. How many of you guys have heard of Earth? Okay. So the rest of you, you're on Earth. Okay, just you know. Now, Earth's a big place. Some places are real hot. Some places are real cold. Where do you think you'd have some kind of mixture where you'd have the highest amount of bacterial diversity? I didn't know this until I talked to my friends who study diversity. Yeah.
Could be the tropics.
It's the tropics, yeah, more specifically the Atlantic rainforest of Brazil, not the Amazon so far as they know. You know, the Atlantic rainforest is kind of this ecotone between kind of this ocean area moving into uplands. So anytime you have variance in what? Habitat, you can have variance in substrate. Remember we talked about substrate variance. And then you have variance going into diversity. And you have a pretty nice temperature regime to work at. So the Atlantic rainforest, is that interesting? The thing about bacteria to think about when you think about diversity is to think about microbial infallibility. You know, this concept that loosely stated is that if there's a natural compound on Earth, there's a microorganism that can degrade it. Okay? And that's why we have pollution. There's things we can't break down because things haven't evolved with it. Now, I'm being very generous. Not reasons we have pollution, but why are things toxic? You know, well, you give somebody a chlorine carbon bond. I didn't grow up with that one. You know, a little bit harder to do it. We see some of the bacteria that can get very creative, like we talked about with PCBs. But microbial infallibility is a cool thing to think about. So look for where there's a diversity of substrate, you'll probably have a diversity of organisms. Habitat's the same way. Okay. This is another thing, is adaptability. Okay. This is against year. As we start looking, we're finding, and this is 2010, we may be finding new domains. Most of you are probably going, I don't care about it. It's kind of interesting. You know, we are on the internet. We know everything about everything in the world. You know, when we touch our screens and stuff. But man, you know, there is just stuff that we don't know about. And who cares? Well, I don't know. I find this interesting that right around 2010, we had a guest lecturer here from Michigan State, and he was telling us about strain 121. And now they've actually named it. You can incubate it at 121. So that still beats out Pyrococcus and Sulfolobus and some of those guys. It turns out that it will exist. It won't die. You can grow it up to 130. I didn't have it. I was too lazy to make degrees. 130 degrees Celsius. Holy smokes. Okay, so as we look, we'll find more. No other group of organisms, though, has the diversity that we've tried to cover in this class. So just showing you the range of things, and where we have this trophic nature. Thermophilic phototrophy, you know, at these temperatures, organotroph, now we get into the lithotrophic life up to there, just because of what is still available and what will work when you start heating stuff up. Again, thermodynamics, physiology seems to be a big part of it. I think we were getting towards the instability of, so how can you exist at least at high temperatures? Some of the stuff we can kind of reverse for low temperatures, if you want to be general. Stability of monomers, protective effect of high concentrations of cytoplasmic solids. Okay. We can put salts in there. We can make things like basically the equivalent of antifreeze. Yeah, you can put water in your radiator. It will cool your engine, but when it gets too hot or too cold, other compounds help to dissipate heat or deal with freezing in different ways. Cells do that. Okay, so solids can be helpful. Use of more stable molecules, for example, use of non-heme iron proteins instead of proteins that use NAD and NADH. So different adaptations on the theme. Don't be intimidated. If you know the blueprint, you know the pattern of how cells work and how physiology and some of this stuff is, it's very easy then to take your scheme and move into the unknown and to understand these things. So this can work for us. Non-heme protein folding is a big deal. I don't have to tell the chemists this, they of course know it, and biologists know this too. Amino acid composition is similar to that of non-thermostable proteins, but you tend to have the folding differently, more hydrophobic cores, okay, making the protein more stable, but then also the amino acids that have ionic interaction, more of those to stabilize the protein is seen in it as a general adaptation here. This go forward? No. High intracellular cellular, we talked about that. Having a different way of folding DNA helps. We didn't talk about gyrase too much in this class, but the way that you fold, when you make that nick and you can unfold DNA in order to replicate it and it folds back up, you can turn the DNA another way or a different way, that can help stabilize the DNA. High intracellular levels of other materials stabilize DNA and RNA. DNA binding proteins show up. Interestingly, archaea have histones. I mean, bacteria are shown to have now like kind of quasi-histone-like proteins, but bacteria or archaea have histones like more like us. What are histones again?
It's the proteins that DNA wrap around to.
Yeah, they're basic. They're full of lysine and arginine and histidine, so they can interact with DNA at neutral temperatures, and they make these nucleosomes. And they're very conservative. It works. Histones work. The H2B and H2A histone only has one or two changes in it between the histone that we have of that and like the garden pea. As long, through evolution, it's been one of the most conserved ones. So it's worked. The archaea have those also. I found that interesting, and they knew that from an early age. And I studied histones for my master's. I always found that interesting. Lipid stability. Okay, we've talked about. Go back, look up those words. If that is like blah 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 to you right now, but we talked about how you can not have a bilayer, but a monolayer. That was kind of an interesting idea. A higher G to C content, triple bonds versus double bonds. Okay. Hard to see if that works with the gram-positives, the Tenurides and the Firmicutes, but with this, it seems to be that. So I think what I did here was simply summarize everything in one place. So that's what that is for you on that. So, okay, I think that's the end of our little discussion of archaea. Sorry if that was a little bit faster than we usually do, but also kind of pruned it down so that you're not overwhelmed on the last day, I hope.